Hi, today we'll be talking about DNA fingerprints, which is used for convicting criminals. The two main analyses that molecular biologists use in DNA fingerprints are restriction fragment length polymorphism and short tandem repeats. Method used by molecular biologists in which crime scene investigators can obtain blood, semen, urine, hair, saliva, and other evidence, known as evidence sample, that can provide a DNA profile. A reference sample such as blood from a suspect, and by the way, 1 nanogram of DNA is usually a sufficient quantity to provide good data. Then, the use of restriction endonuclease enzyme to cut up DNA into different lengths, separating the DNA fragments by a gross gel electrophoresis, determining the number of fragments and relative size, which is ultimately used predominantly by DNA laboratories until newer technology was developed. In the past, it could take as long as a couple of weeks to obtain results using RFLP. It also requires the use of a sizable amount of DNA. The second method is short tandem repeats, which forensic DNA started to use in 1991. In short tandem repeats, we need biology to extract and discern DNA quantity and to use PCR for amplification of multiple STR markers. But the use of STR is becoming the preferred method of analysis because of its accuracy and most importantly because STR uses shorter strands of DNA, while RLP require a large amount of undamaged DNA. Therefore, the FBI has chosen 13 specific STR loci to serve as the standard for codings. When a DNA polymerase encounters a repeated sequence, it has a tendency to slip a little, leading to increase and decrease in the number of repeats, which is a source of polymorphism. By determining the sequence that margin the STR and amplifying them through PCR, which stands for polymerase chain reaction, the difference in length can be detected by gel electrophoresis. We also need technology to separate and distinguish the repeated sequence that was cut by PCR. And finally, we use genetics to compare samples of genotypes to other sample results. In other words, STRs are short segments of DNA that are repeated a number of times in a sequence. The second method is short tandem repeats, which forensic DNA started to use in 1991. In short tandem repeats, we need biology to extract and discern DNA quantity and to use PCR for amplification of multiple STR markers. But what is CODIS? CODIS stands for the Combined DNA Identification System. It was officially activated in October 1998. Allowing participating forensic laboratories to compare DNA profile with the goal of matching a case evidence to other previously unrelated case, or to person already convicted specific crime. Although the CODIS proved its importance, some people oppose it, saying that the indexes will include the DNA profiles of innocent individuals, violating the right to privacy. There is also a risk associated with the Combined DNA Identification System, which occurs if the Combined DNA Identification System contains profiles that are inaccurate, incomplete, or unallowable. In this case, the database is less useful to the criminal justice system. In addition to convicting criminals, DNA fingerprinting has been used to free wrongfully convicted people. In 1984, Darryl Hunt was sentenced to life in prison for raping and murdering Deborah Sykes of Winston-Salem. He was just 19 years old, but he was also African American, and Sykes was white. Although there was no physical evidence linked Hunt to the crime, but there were claims made by a hotel employee, later proven to be false, and he said that he saw Hunt enter the hotel bathroom later that morning and exit leaving bloody hand towels, as well as other witnesses who placed Hunt near the scene, and that's why he was accused of a crime that he did not commit. He was in prison for almost 19 years. His lawyer requested a DNA test for him, and in 1994 the results came back, and his DNA did not match the semen found on the victim's body at the crime scene. Despite the results, Hunt's appeals were rejected. Judges found that the new evidence did not prove innocence, and he served another 10 years. But finally, in 2004, the DNA profile from the crime scene was run in the state database, at the request of Hunt's attorneys. The results matched a man incarcerated for another murder. Of course, without the Combined DNA Identification System, they wouldn't be able to match the DNA with the other criminal who truly committed the crime. Hunt was exonerated and freed in 2005. In Darryl's case, probably his DNA was analyzed with the method of short tandem repeats. After his exoneration, Darryl established the Innocence Project, as well as his own group called the Darryl Hunt Project for Freedom and Justice. This project is devoted to educating the public about flaws in the criminal justice system, advocating for those wrongfully incarcerated as a result of those flaws, and providing resources and support for those trying to rebuild their lives. Science has a great impact on our life. It is the answer to everything, and as we live, we get introduced to many developments and improvements in science that makes our life easier day by day. For the DNA fingerprint, a lot of crimes wouldn't be solved without it, and a lot of people like Darryl Hunt would remain in prison, although they are innocent. And for the combined DNA identification system, I think that it should be provided for every country to maintain a peaceful life.